Everyone, today I am going to explain about the structure of bacteria and its types based on the position of the flagella. So, at first let us study the ultrastructure of bacteria. Electron microscopic studies have revealed the ultrastructure of the bacterial cell. The outer envelope is the cell wall, internal to which is the cell membrane. Sometimes external to the cell wall there will be a loose slimy layer or capsule. Some bacteria are flagellate, others have small pili or fimbriae. The bacterial cytoplasm includes ribosomes, mesosomes, fat globules, vacuoles, inclusion bodies and nuclear material. So, in this picture you can see the ultrastructure of the bacteria, that is having flagella as well as cell wall, cell membrane, cytoplasm, capsule, plasma membrane and also nuclear area, nuclear material and DNA, plasmids, fimbriae. So, these things you can see in this picture. So, let us study one by one, that is capsule. So, in many bacteria, that is Diplococcus, Pneumococcus, a viscous or gelatinous substance is secreted surrounding the cell wall, that is called as capsule. Usually capsule are thick and sharply defined. In some cases capsule are too thin and they are called as microcapsules. In Leuconostoc, the viscous substance is very loose and undemarcated and is known as slimy layer. Chemically, the capsules are made up of proteins, polysaccharides and lipids. Some slimy layer polypeptide are built with a single amino acid. The capsule may not be present in all the bacteria. And the next one is after the capsule we can see the cell wall. The cell wall is thin, about 10 to 25 nanometer thick, and it provides rigidity to the cell. It is visible only under the electron microscope. So, let us say the chemical structure of bacterial cell wall. Okay, chemically cell wall consists of mucopeptides made up of alternating chains of N-acyl glucosamine and N-acyl muramic acid molecules. These are cross-linked by peptide chains. The narrow space left by these chains are filled by various other chemical compounds. The cell wall composition differs between gram-positive and gram-negative bacteria. The cell wall of gram-positive bacteria contains up to 95% peptidoglycan and about 10% teichoic acids. The gram-negative bacteria, the cell wall composition is more complex. It is made up of several layers. So, next to the cell wall we can see the cell membrane. Internal to the cell wall is the cell membrane, or it may also called as plasma membrane. It forms the outer boundary of the cytoplasm and is selectively permeable, and thus it regulates entry and exits of molecules into the cytoplasm. The membrane is chemically made up of lipoproteins, that is in the ratio of 70 to 30 ratio, that is 70% of proteins and 30% of lipids, and it does not contains any kinds of carbohydrates. So, in the cell membrane we can see the absence of carbohydrates. Electron microscopic studies have shown that the membrane is a three-layered one with a unit membrane. There are two electron dense layers encompassing a central electron transport layer. The outer layer are about 3 to 5 nanometer thick and the middle electron transparent layer is about 5 nanometer thick. And in this cell membrane we can also see the lipids. So, lipid found in the membrane are phospholipids such as phosphatidyl ethanol amine. The bacterial cell membrane performs the following functions. They functions as a osmotic barrier and it contains an enzyme system involved in the biosynthesis of membrane and cell wall components. Contains components of energy generation system. And next to the cell membrane we can see mesosome. In some bacteria, particularly in the case of gram positive bacteria, the cell membrane forms vesicle or packet like enfoldings into cytoplasm at several regions. These are called as mesosomes. Mesosomes are the reservoir of respiratory enzymes. So, next one is cytoplasm. It is the viscous substance. Cytoplasm includes ribosomes, nuclear materials, proteins and other water soluble materials and reserve food material. Organelles like endoplasmic reticulum, mitochondria, Golgi bodies are absent and plasmids and episomes are also found in cytoplasm. So, in the next thing it is ribosomes. These are dispersed throughout the cytoplasm and as in eukaryotes are involved in the protein synthesis. The number of ribosomes are per cell varies and may reach up to 15,000 per cell. Each ribosome is a ribonucleoprotein particle of about 100 to 200 Armstrong units in diameter. Bacterial ribosomes are of 70S type as noticed by their sedimentation properties. So, next one is nuclear material. The nuclear material is dispersed in the center of the cell. It is called as nucleoid because it has no nuclear membrane or it does not contain nucleolus. So, nucleoid consists of closely packed fibrillar DNA. Studies indicate that it is a single molecule of double stranded DNA. RNA may also associated with DNA. The bacterial DNA molecule is often referred to as the bacterial chromosome. And the next one is plasmids and episomes. So, in many bacteria, DNA is present outside the chromosome, that is outside the nucleoid. Such extra chromosomal DNA is referred as plasmids or episomes. It is difficult to distinguish between the two and they are often regarded as synonyms. Plasmids are drug resistance, toxin producing ability, etc. The size of the plasmid is about 1/10 of nuclear DNA, but a bacterial cell may have one too many plasmids. Plasmid DNA is also double stranded and circular. So, the next one is cellular reserve materials. A variety of reserve food materials are found in the bacterial cells of these. Starch, glycogen and poly and hydroxybutyric acid are important among the cellular reserve material. And the next one is flagella. So, flagella are long thread-like appendages attached to the cells found in bacilli and spirillum. They bring about locomotion in the moist medium. The flagellum is thin and about 0.02 micrometer in diameter, hence invisible under light microscope. Each flagellum is about 3 to 20 micrometer long and terminates in a squarish tip. Electron microscopic and X-ray differentiation studies have shown that the flagellum is made up of three parts, that is filament, hook and basal body. Filament is made up of protein fibers and about 13 to 14 nanometer in diameter. It is outside the cell and connects to the hook at the cell surface. Hook is bent and is thicker than filament, that is 17 nanometer, and it is penetrates the cell wall. The basal body is a spindle shaped structure that joins the hook to the cell membrane. A short collar like structure attaches the basal body to the hook. In gram negative bacteria, the basal body has two pairs of rings. The outer pair, L and P rings, is fixed to the cell wall with the inner pair, that is S and M rings, that is L and P rings that will pairs with the inner pairs, that is S and M rings, that is attached to the cell membrane. In case of gram positive bacteria, the basal body is much simpler and it has only one ring. So, the next one is fimbriae or pili. These are short, very fine hair-like processes found in some gram-negative bacilli. That is, they are also called as pili. They are about 0.5 micrometer in length and less than 10 nanometer thick. They originate from the plasma membrane and their function is to adhere to the cells, particularly during conjugation, and so that these pili are also called as sex pili. So, this is about the ultrastructure of bacteria. So, the next one is types of bacteria based on the flagellar distribution, or the on the basis of the position of the flagella, the bacteria are divided into 5 types. Okay, that is, except the coccus, all other forms of bacteria may possess organs of locomotion, that is flagella. The flagellum is a long thread-like structure whose length usually exceeds that of cell. It arises from a basal granule. On the basis of number and position of the flagella, bacterial forms are classified into 5 types. In that, first one is Atrichus. In the Atrichus kinds of bacteria, the bacteria doesn't contains any kind of flagella. Example is Coccus. And the next one is Monotrichus. In the case of Monotrichus, single flagellum is present at the one end. Example Vibrio, you can see in the picture. And the next one is Lophotrichus. Many flagella are present at one pole of the bacteria. Example Spirillum. And Amphitrichus. Two tufts of flagella are attached at the either poles of the cell, that is spirilla. And the next one is Peritrichus. Flagella are present all around the bacterial cell. Example Salmonella typhosa. So, this is about the based on the position of the flagella, how the bacteria are classified. So, thank you.